Recording started.
All right. Yeah, and by the way, I hope I can see everything on the screen okay today, because I've got the sun shining in the window, and my window is really frosted up, and you get this kind of diffraction effect going on. You get refraction, you get diffraction of the light and all that kind of stuff, so it's quite bright shining in here. So just in case I'm not able to see something on the screen for a bit, that's why. Yeah, it's pretty wild. So, so everything's going well? Getting ready for Christmas, David? Oh, good stuff. Right on. And I don't know if I mentioned to you, did I tell you that we're getting a little puppy dog? We're going to be buying one soon. You're done your wrapping as well, eh? Man, you are quick this year. Are you always this quick? $2 a.m. on Christmas Eve, so it's just a really quick one this year, eh? Good stuff. That's cool. Okay, so I'm just going to get on the first page here. So basically it's talking about a population sample, and you're probably familiar with this from the idea of, you know, when you're doing an election, you take a sample of the population and it represents the whole population. And so it's the same kind of idea when you're talking about population sampling with organisms in an ecosystem. So just to review again, when you're talking about a population, that's specifically talking about a group of the same species living together, whereas a community is when you have all different kind of species. So you might possibly have, you know, lions and tigers and maybe antelope, and together they might make up a community, whereas a population, when it's just one species, that's, you know, for instance, a bunch of finch birds or a bunch of the same specific type of plants or whatever. Okay, so you'll find these first ones really easy, I'm sure. So question one, a group of organisms of one species living in a particular habitat at a certain time is called, what would you call that? K population, good stuff. Question two, blank is made of the populations of all organisms that occupy an area. So in other words, a number of different populations, number of different species. Okay, that's community, good stuff. Okay, now here's just the point that populations tend to stay stable over time, especially if they're larger populations. And so we've got this principle, it's called the Hardy-Weinberg principle, that says that alleles and genotype frequencies remain constant or stable as long as 5 conditions are met. Okay, so what are those 5 conditions? So the first one is that populations are large enough. And later on we'll look at what happens if a population is small. It's kind of an exception to the rule here. If mates are chosen at random, of course that's not normally the case. But these are all assumptions that you need to make in order for the Hardy-Weinberg principle to be, you know, working absolutely perfect. The next one is no net mutations, no migrations. Of course, again, these are all assumptions. And the last thing is no natural selection against any phenotypes. But again, these don't all hold true. Okay, but those are the 5 things we need to assume in order for the Hardy-Weinberg principle to work perfectly, at least. Okay, so we'll go on to the next point here. So the Hardy-Weinberg principle is represented by this quadratic equation. I don't know if you've seen this before. Basically, we usually represent P as the frequency of one of the alleles, and we usually choose P for the dominant allele. It doesn't really matter that much because P squared or Q squared, they're both doing the same thing in the equation. So Q represents a frequency usually of the recessive allele. Okay, so if we imagine a gene that has capital A and small a, if the frequency of capital A is 0.7, then the frequency of the small a is going to be 1 minus the frequency of that. So it's always 1 minus whatever the frequency of the other allele is, because together the percentage of both alleles is going to be 100%, right? And 100% is represented by 1. So if you look at it here, if you were crossing big A by big A little a, then these are the different possibilities you get, and these are the probabilities that would go along with them, right? Sorry, I just need to get the phone really quickly. I'll be back in just a sec, David. So if you just want to look at that for a sec. Okay, I'm sorry about this. I just have to fire off a quick email, and then and I'll be right back. Sorry about that, David. Okay. Yeah, so where were we then? So the way this plays out then is if you look at those probabilities that we looked at, so here's the formula. So we have p squared plus 2pq plus q squared. Since we said the probability of one of the alleles was 0.7, okay, that was a capital A, and again, we usually just attach that with p, that the dominant allele is p. So we make that 0.7 squared, and since it's 2 times p times q, so it's, the probability is 1 is 0.7, and we know that because it's 1 minus the probability of 0.7, it's going to be 0.3 for q, and so that's how we get this calculation. And then for q, it's going to be 0.3 squared. Okay? So then we do the math, and for p squared, we get 0.49. For 2pq, we get 0.42, and for q squared, we get 0.09. And what those probabilities actually mean is that the probability of getting AA, in other words, 2 dominant alleles, is 49%. And we get that from the 0.49. Okay? So that's what the p squared actually means. When we find it, it's the probability of getting both dominant alleles. And then q squared is going to be the probability of both recessive alleles, and we see that's 9%. And then this here, this 0.42, is going to be the probability of getting a dominant and a recessive allele. Okay, and we also, if we look at the Punnett square, we see we have 0.21 and 0.21, and when we add them together, that's 42%. Okay, so that's what that formula, how it actually works out when we're doing kind of calculation problems with the Hardy-Weinberg principle. Okay, so do you have any questions about that? Yeah, you could, it would probably be good, especially on the diploma exam, to just first write it out just to show what it looks like, sort of thing. And then after that, if you wanted to switch it into, a capital A, little a, that sort of thing. Okay? Because they might be looking for that as just part of the solution. But yeah, the most important thing is that you can use it and to get the different probabilities. Okay, so shall we just go on to the next question? Okay, so the next question says, a technique in which gene frequencies for a particular genetic trait are determined in a small sample of the population and results are applied to the whole population is known as, so what do you think the answer would be for this one? That's right, that would be population sampling. And the next question, which of the following indicates conditions under which allele and gene frequencies will remain constant from generation to generation? That's right, good stuff. And the next question, if the frequency of allele A is 0.9, and the frequency, okay, that's capital A, the frequency of small a is 0.3, what percentage of the genotypes would be big A, little a? Okay, so this is kind of similar to the last question we did, except we've got different numbers here. So what do you think it would be? Sure thing. Okay, 54%, good stuff. All right, so I'll just move on to the next page. Screen number 4. Okay, so this one was talking about that chi-squared stuff, or x-squared stuff. And so basically the purpose of doing the x-squared is in testing the discrepancy between a set of observed or experimental results and theoretical results. So in other words, you think you might get this, but instead you get this. So when that's the situation, you apply this x-squared system of running through the results, and by that you're able to find out if the experimental results are within the realm of what you would expect for theoretical results. In other words, are your results likely to be due to chance, or are they likely to be due to something else? So for instance, if you were doing some tests and you're finding out that you've got certain results, you get a certain percentage for this x-squared, well, a certain number for the x-squared, you plug it into the table and you get a certain percentage. Let's say it's trying to discover the correlation between the number of storks in the area and the number of babies in the area. And you find out that you get a certain percentage, and it says that, okay, the correlation between number of storks and number of babies is actually due to chance. It's not a significant correlation sort of thing. Whereas if you're getting something, if you're doing a test having to do with how much water you give to plants and how much growth they have, you're likely to find that the results are what's called statistically significant, right? So that's kind of what this is looking at. So if you get a percentage of 5% or less after doing the x-squared thing and putting it on the table, what that means is that the difference is probably due to chance alone. So that's the type of result you would likely get if you were doing an experiment with storks and babies. You're going to get a result that's 5% or less. Okay, and so go on the next screen. Okay, so here's the table. Here's how you would use it. And so if you have two variables, then you're looking at two classes. That's kind of what it means by two variables. And so you take the x-squared value that you're given, and here it's showing you, I know this table is not as clear as it could be. Here it's showing if you were to get 5% or 1%. So those are the kind of results you'd get if it's due to chance alone. Okay, so when the probability is less than, okay, so I'm sorry, I didn't tell you the right information. The larger number is when you're getting a result of chance alone. And that includes 5% or greater. Okay, if you're getting a result like, say, 1, so in other words, if you get a low percent, then the results are said to be significant. And the difference did not occur by chance alone. So we're looking at the difference between the expected results and the experimental results and the theoretical results. And we're saying this is something that's significant that's going on, and it's not due to chance alone. There's actually something taking place that's scientific that's going on here. Okay, so if you got a result like 1, then you would say, hey, something's going on here. We're not looking at the stork and baby effect. We're looking at the watering plants and growth effect. Okay, so that's basically what that's about. And so if we go on to this question here, this says when a chi-squared, so we either talk about it as chi-squared or x-squared. X is kind of chi in the Greek. So when it was calculated and a value of 1.422 was obtained, and we looked at a probability on the table for 2 classes, we found it to be significant at the 5% level. Okay, so what does that mean then if we found the results to be significant? Which of those answers would be correct? That's right, it's not due to chance. Okay, good stuff. So we'll go on to the next concept here. It's talking about genetic drift. And genetic drift is basically when you're talking about small sample sizes. So a small population, rather, I should say, what happens is you get a deviation from what you get with the Hardy-Weinberg principle. So what can happen if you've got a really small population is the dominant gene may disappear from a small population. Because the organism if carrying the gene may fail to reproduce before it dies, or it may fail to reproduce, or it may die before having the chance to reproduce. So when that happens, you call that genetic drift. So you get a different effect going on with the population than you would if you had a large population. Okay, so the answer for question 7 then would be, what would it be? Which of the following explains the disruption of the genetic equilibrium in small populations? Okay, good stuff. So, on to the next board. Okay, so the next board is talking about 2 concepts, geographic range and ecological niche. So the geographic range is the region where members of the species have been sighted. Okay, so you're talking about the whole large area that this particular species have been sighted. That's the geographic range. And a niche is what we talk about. It's the role or function played by a population in a community. So, for instance, is this creature a producer? Is it a consumer? Is it a predator, prey? But usually when you talk about the niche, it goes into a whole range of different things. It's like, what creatures does this creature eat? Who does it get eaten by? Where does it live? Well, mind you, that's kind of habitat, but it kind of figures into the whole, what is the niche. There's a whole bunch of different things that go into, what the creature's role is in the particular environment it lives in. So that's kind of what its ecological niche is. So for question 8, we would say blank is a region where a given organism is sighted. What would that be? Okay, next question's ecological niche, right on. 2-for-1 deal there, eh? Great stuff. All right, so the next the next topic is looking at population patterns. And when we look at different population patterns, so we're looking at the different kind of growth patterns that we can see when populations grow in an area. So if we were looking at, from say, a bird's eye view, what would it look like if we saw this population distributed? So we can have 3 different patterns. We can have clumped, random, or uniform. And we can call uniform, we can also call it spaced. So a clump distribution is basically, like the name suggests, you get clumps of organisms living. You've got spaces with no organisms, and you've got clumps, or maybe you've got just a few in an area, but maybe not forming a clump. But for the most part, you're seeing clumps of organisms together. And usually what causes them to clump is when you have, sunlight in a certain area or something like that, or moisture in a certain area where they can get water, then that's what's causing the clumping. Random distributions are are not very common. So usually you either get clumped or uniform. And neither biotic, in other words living, or abiotic, in other words non-living factors, have much of an effect on random distributions. So an example is, people milling around in a mall. That's an example of a random distribution. Probably most malls you wouldn't see random distributions. But anyways, okay, so we got some pictures here, and the last one is uniform distribution. Okay, so that's uniformly or evenly spread out. And so when there's competition among individuals for things like food, water, space, and light, okay, you might have a uniform distribution in the classroom. So here's what it looks like, a clumped distribution. You can see the clumps. Here's evenly spaced, and here's random. Random looks kind of evenly spaced, but it's kind of a combination between clumped and spaced, I guess you could say. So can you think of any example, David, of plants that would exist in a uniform or spaced distribution? Okay. Yeah, trees on a tree farm. Good idea. Or also another one might be grass, right? Grass is pretty uniform on a lawn. Might be a few areas of clumping, but it's pretty uniform. How about clump distribution? You think of some plants that would exist in that kind of distribution? Oh, what was that? Okay, the soup-weed yucca. Okay, good. I'm not that familiar with that plant, but yeah, like if you had trees occurring in a field, it wouldn't be surprising to see them. Oh, soapweed. Okay, cool. I'll have to check that out. Yeah, or also if you had plants in a field, a lot of times you might see them kind of forming some clumps based on the amount of light available or the amount of moisture available, right? If moisture would kind of pool in an area, that sort of thing. Okay, so question number 10 here says, distributions which are arbitrary and appear to be unaffected by biotic factors are said to be, which one would that be? Random. That's right. Good stuff. Okay, so here we're talking about population size and population density. So population size, of course, is the number of organisms of the same species sharing the same habitat or space at a specific time. Population density is the number of organisms per unit of time. So density equals n for number divided by S which is the population size. Okay, so we'll go on to the next screen. Okay, I'm sorry, population size, what am I saying? That's the space occupied, because you're looking at the number in a certain space. So it should be D equals N divided by S, number divided by space. Okay, so the rate of change is also an important concept. So rate of change of a population is equal to change in D over change in T. And change in D is change in density, change in T is change in time. So whenever you have a change in something, it's the final something minus the initial, whatever it is. So if you're looking at a change in temperature, it's final temperature minus initial. And so we're looking at the change in density divided by the change in time. So we've got a question here, just an example. So the population density in Edmonton goes from 100,000 in 1959 to 500,000 in 1995. And so what we do then, we subtract one density from the other, and we subtract 1 year from the other. Okay, so our change in density is going to be 500 minus 100,000, which is going to give us 400,000, and then we're going to be dividing by 36 years. So we'll go to the next page to see that continued. So we can see, well, just got kicked off the stool, just going to check here. Okay, so on the next page, what happened was, because we multiplied by 100, we just, rather than this, having this as 100,000, at one point this was crossed off. So this was 400,000, and that was crossed off with the 100. And so that's why we just have 4,000 at this point. Okay, so we've got 4,000 divided by 36, and that comes out to 111 people per kilometer squared per year. So the number of people per kilometer squared, that's the density, and then it's per year. Okay, so we know that if R comes out to be positive, the population is growing. If it's negative, the population is getting smaller. Okay, now if R is really large, the population is either growing or shrinking rapidly. Okay, so if it's a really large positive or really large negative, then it's either growing rapidly or shrinking rapidly, and if it's small, then it's either growing or shrinking slowly. Okay? So, for question 11, the change in population over a period of time is known as, what would that be? Okay, rate of change, good. And if we have a positive R, that means the population is what? Growing, good. All right. So, next screen, we're talking about 4 different factors that affect the change in population sizes. So we can either have natality, which is another word for births, immigration, so people are coming in and joining our group. We can have mortality, which is another word for deaths, and emigration is another name for individuals leaving our group, leaving the population. So we can see that both natality and immigration add to or increase the number of individuals of the species, and both mortality and emigration subtract from or decrease the number of individuals. So if we want to find the percentage change in a population over time, you can see what we're doing here is we're adding birth and immigration in this bracket, we're subtracting death and emigration, and then we're multiplying by 100 because we've got a percentage, and then we divide that by n. Okay, so n is just the number in the population. Alright, so that's what we do to find the percentage change in a population. We use this formula here. Alright, so going on to the next screen, here's an example then. We've got 200 white-tailed deer, we've got 20 born, 10 die, and then we have 5 move into the park, So that 5 is going to be immigration. 2 leave the park, so those 2 are going to be emigration. So the things that are causing it to go up are the 20 that are born and the 5 that move in. And then we subtract off the 10 that die and the 2 that are leaving the park. And then when we plug it into this formula, we're eventually going to get 25 minus 12. We get 13 per year divided by the 2. Okay, and then remember we had 200 here. And what we did is we had 200 on bottom, 100 on top. We crossed that off, and that's why we've got 2 on the bottom. And so we've got 6.5% per year. All right? And let me know at any point if you want me to go over something again. So this here, this concept is talking about mature ecosystems are fairly stable over time, right? So if you've got a mature forest, you've got a fairly stable ecosystem over time. And so we call this a steady state or dynamic equilibrium. The reason we call it dynamic equilibrium is because you've got some creatures leaving and some staying, but overall it's roughly equal. Okay? And that's why it's dynamic. Dynamic is changing, but equilibrium means it's equal. Okay, so so we do have some natality, some death, and some mortality in a dynamic equilibrium. Okay, so in a natural or open population, normally there's no immigration or emigration in a closed population. So, okay, so if you're looking at something like a farm herd, okay, that's a closed population. Okay, so equilibrium means that the net size of the population remains the same. So if we go to the next screen, okay, so a blank is one which density changes are the result of natality and mortality with neither food nor waste being allowed to enter or leave the given environment. So what do you think that would be? Okay. This one, I think this one's a little bit of a tricky question, the way it's worded. This one was actually looking for dynamic equilibrium because it's looking at the fact you've got both births and deaths. It should have talked about births and deaths occurring at the same time. But I believe that this one was looking for dynamic equilibrium. I'm going to check this one over again, though, because I can see how you could say it was a closed population as well. So I'm going to check into that one, David, because, yeah, I can see. Okay. How about I just have a look at this one again and maybe get back to you on this one again? Is that okay? Because I think I might have missed presenting some information here. Okay. Yeah, maybe if it's okay, David, I just want to check into something. And I'll send you an email later on if that's okay. All right, great stuff. So we'll just kind of put that a little bit on hold for now. At this point, we're going to talk about biotic potential, which is stood for represented by Rmax. So Rmax is the theoretical maximum number of offspring produced by a population under ideal conditions. Okay, and now there's when you're looking at the word environmental resistance, that's all factors that tend to reduce population size. They can be either living or non-living, such as climate change, competition, disease, and predation. Okay, so when we look at the term biotic potential, what that relates to, so again, it's the maximum number of theoretical offspring produced by a population. And it's related to fertility, so it's related to a particular species' ability to produce offspring, and also what's called fecundity, which is the number of offspring per birth. Okay, so that's a little bit different than fertility. Fertility is the ability to produce offspring, and fecundity is the number of offspring per birth, and the frequency of births. Okay, so those three things have to do with biotic potential. So 4 different factors have to do with, they regulate biotic potential. The number of offspring per birth, the maximum number. Okay, so if you're looking at a rabbit, it's going to have a lot of offspring. Capacity for survival, so I mean you might produce a lot of them, but if they don't live very long, that's going to decrease your biotic potential. The procreation rate, so the number of births per unit of time. Okay, so in other words, what's the for humans, the 9 months is the kind of the limit where the amount of time whereby you can produce more children. And whereas for if you're looking at something like a bacteria, it's 20 minutes, produce children every 20 minutes. So maturity age is the age at which offspring are first able to reproduce. So all those things have an impact on the biotic potential. So we're going to look at the 2 main types of, I guess in just a little bit we'll look at K-selected and R-selected. First we'll just look at a few other concepts. Okay, so if you're looking at something called a limiting factor in a population, that's all the different factors required for growth. But there's one factor that has the lowest relative level. That's the controlling factor, or that's the limiting factor. Okay, so if it's, for instance, it might be the amount of food that you have, it might be the limiting factor for a population, or it might be something else. Okay, it's kind of like in chemistry they have something called the rate determining step. That's the slowest step in a series of chemical reactions. And whatever the slowest step is, it determines how fast the reaction is going to go. That's kind of related to what a limiting factor is, or it's kind of analogous, I guess you could say. So anything that affects the size of a population is called a what factor? Okay, controlling factor or limiting factor, right? And so when we're looking at populations, we normally see what's called an S-curve. So if we imagine, just what an S looks like, that's what it's going to look like. And there's 4 different parts to it, especially when you're looking at bacteria, you see this kind of thing going on perfectly. You've got the lag phase, the growth phase, the stationary phase, and the death phase. So the lag phase is when the members of the population are adjusting to their new environment. So it kind of takes them time to get going. And they don't start growing right away. And so in the beginning, the growth is slow. That's the lag phase, just like the word sounds, right? Lag, and it sounds like it's kind of a slowdown, right? The growth phase is what happens to a population under ideal conditions. Okay, so you're looking at a few deaths, lots of nutrients, not much waste, no overcrowding. That's what happens in the growth phase. And that's what you're talking about. So when you're talking about the growth phase, you're also talking about exponential growth. Okay, the word exponential means things are really, really growing. And it has that math kind of suggestion as well, right? It's like when you go 2, 4, 8, 16, that's exponential growth. So the stationary phase is when growth kind of slows down to a, it's more just a straight line. It's not going up and up and up. And then the death phase is when the growth is actually going down. So if you see what it looks like on a graph, on the next screen, here's the lag phase. There's not a lot of growth because the organism is getting used to its environment. The log phase, or growth phase, or exponential phase, is when you got a lot of growth. Then the stationary phase, you can see things are really slowed down. You've got some overcrowding. You don't have enough nutrients for everybody. Things slow down. And the death phase, eventually things slow down so much and nutrients run out, and so we get that sort of situation going on. So you can see it's kind of an S pattern, right? All right, so we'll get back to that in a little bit more later on. But first of all, we'll look at, when we talked before about biotic potential, we've got K-selected populations and R-selected. So the main important thing to connect with this is that K-selected populations are going to be creatures that are large creatures. So if you're going to look at something like a horse or a cow or a bear, whatever, those are going to be K-selected populations. So let's have a look here at the different characteristics of their population. It's going to be a stable population. There's going to be few but predictable fluctuations. Populations are going to be crowded. You know, if you've got bear living in Banff, they need a large ranging area. They experience what's called intraspecific competition, so between the species members, between the other bear, not between bear and some other species. And only the best adapted adults are able to reproduce. Okay, so it's the population size is close to carrying capacity, and again, they're large. They grow slowly. The young require a lot of parental care. That's another really important characteristic of a K-selected population. Reproduction rate of individuals in the population is low, and the young have the poorest survival rate. Okay? So we'll contrast that now with an R-selected population. If you want to remember R, think rapid growth. R starts with an R. These are small creatures, could be something like an insect, bacteria. They have short lifespans. Reproductive rate is high. The young are precocious. I'm not even sure what they mean by that exactly in this context. Anyways, I guess what they mean is they reproduce at a young age. I'm not sure. Anyways, the adults are the most susceptible to rapid environmental changes. Okay, so we can see some differences between R-selected and K-selected, but some of the main differences are the size of the creatures and things like the lifespan of the creatures. You know, a bear has a very large lifespan, an insect has a short one. Insects reproduce at a young age, bears at an old age relative to how old they live. So, okay, so question 15 says, what's the most common type of growth curve seen in populations? So what would you call that type of growth curve? Okay, it's the S-curve, and can you remember what the four parts of it are? Okay, lag, log, stationary, and depth. Good stuff. And the lag and the log, they kind of sound similar. And when you think about it, they sound similar, lag and log, and they both kind of represent growth, or more, you could say more aggressive growth. And the last two, stationary and depth, kind of represent more things are winding down. So, good stuff. And then the four, what would you say are some of the major differences? Sorry, I don't mean four. What are some of the major differences between K-selected and R-selected populations? Okay, lifespans, regular creator size, and reproduction rates. Good stuff. All right. So, we'll just talk briefly, we kind of touched on this a little bit earlier, intraspecific and interspecific species competition. And so Gauss's principle says that if two populations of organisms occupy the same ecological niche, one population will be eliminated. And again, these principles, are principles that kind of are not perfect, but things tend to happen that way. If you have two species in the same area, or two of the same species of bear or whatever, the two populations, say, one comes into another area, one of them tends to be eliminated. So that's intraspecific. That's between the same species of, say, bear. Interspecific is between different species, and you can also think of it as difference between the intranet or internet. Internet is connecting different computers, whereas we're all on CyberHi on the intranet, which is we're all on the same computer kind of thing. And so that has to do with interspecific competition involves competition between different, though often closely related species. Okay, and so now looking at, you might remember some of these terms we're going to talk about next from earlier grades in science: parasitism, commensalism, and mutualism. They're all types of symbiotic relationships. And symbiotic is just two creatures living together in close association. Some people think of symbiotic as meaning that they always get along, but it doesn't actually mean that. It just means they live closely together, but in some situations they get along and some they don't. So do you remember, okay, so for instance, well, I'll just tell you, the difference between them is who benefits, right? One of these terms is where one creature benefits and the other is harmed. The other is where both benefit, and the other is where one creature is harmed, the other is not harmed, but neither does it benefit. So it's just kind of unaffected. Do you remember, David, which one of these would be a situation where one is harmed, one benefits and one is harmed? Which of these three terms? Okay. And do you remember which situation is the one where both benefit? Okay. I'm not sure what you're reading. Oh, sorry. Go ahead. Okay, that's okay. Okay, so yeah, it's just like if you say the feeling is mutual, right? If you say the feeling is mutual, it's like we both feel the same way sort of thing, right? And we both benefit sort of thing. So that's mutualism. Commensalism is when one creature benefits or one organism benefits and the other one doesn't. So if you're looking at, say, a nest in a tree, right, that would be commensalism because the bird benefits from the tree, but the tree doesn't really benefit at all. It just has the bird there, but it's not really harmed or benefited, right? Whereas mutualism, if you got some creature cleans off the bottom of a shark, it's helping the shark, and it's also benefiting by getting food. So that's like the remora shark. Okay, so we've kind of gone through those there. I'm not going to go into them anymore. And so I'm just going to go over what's called succession. You remember talking about succession? So succession is the gradual orderly change in vegetation also in animals, but usually we're talking about vegetation. We can call them different seral stages. Okay, and it develops to a final stable community called a climax community. And there's two different types of succession: primary succession and secondary. And the main difference between them, primary succession is where no organism has ever grown before. Okay, so if you're looking at something like bare rock, that's where a primary succession is going to take place. And so what's got to happen is, lichens are going to grow on the rock, they're going to break it down, and then that's going to provide a foundation. As soon as you start breaking down the rock, it produces soil, okay, and then eventually you're going to build up to a climax community. And so if you're looking at secondary succession, okay, so secondary succession, it occurs in the area where there has been some soil, but it's been covered over by vegetation. So for an example, a forest taking over an abandoned farmer's field, that's an example of secondary succession. So here's a picture of primary succession. You can see we had rocks, but eventually they're going to break down, they're going to produce soil, you're going to get these small herbs, and then you're going to get larger and larger plants until eventually you've got this climax community at the end. You can see with the secondary succession, we start off with, say, weeds in a farmer's field. We get more advanced plants growing larger and larger until we've got this forest at the end. Okay, so that's kind of the difference between the two. Next stage here. Okay, so here's a few generalizations. The types of species in the community change most rapidly during the earlier stages, and then it becomes stable near the end. Second point is that the total number of species increases dramatically during the early stages, and it levels off as you get to the climax. Okay, so question 17, an abandoned farm would likely be, what type of succession would that be again? That's right, secondary succession. An area uncovered by a retreating glacier would most likely be D, good. And then, I don't think we have any more on this page. Screen number 20. And we've only got, after 21 we're done. So, okay, so succession on bare rock is a slow process because, so why would it be a slow process on bare rock? D, that's right, because the first plants, they're called pioneer plants. And for instance, yeah, I was going to tell you, after a fire, the first plants that come in, I believe, are called pioneer plants as well. Okay, so for the next question, it says for each of the following state the disease that fits with the description. Okay, so what do you think the first one would be? A disease caused by an abnormal form of hemoglobin. And if you can't remember which one these are, that's okay. Okay, this one is sickle cell anemia. You might remember that what happens is you get a genetic problem going on, and what happens is the hemoglobin normally has that kind of specific shape to it that helps it to carry the oxygen. But what happens when you get sickle cell anemia is the hemoglobin no longer has that shape because of the mutation, and so because of that, the hemoglobin, the red blood cells get kind of clumped up because of that, and so they're not able to carry the oxygen as well. So that's sickle cell anemia. How about for B? Do you know which one that would be? A sex-linked disease that can cause fetal bleeding and follows an excellent inheritance pattern, and homozygous individuals are dominant. In other words, you get a different sort of a pattern going on there. If you've got two recessive, if you've got a creature with two alleles, two of the disease alleles, they're going to be dominant rather than recessive, which is normally the situation. Do you know which disease this would be? Excuse me. Yeah, I bet once I tell you, you'll recognize it. This one's hemophilia. So again, hemophilia is fetal bleeding. It's an excellent pattern, and I remember we looked at it when we looked at some of the pedigree charts. And in this situation, homozygous individuals are dominant. Okay, the next two are probably going to be tricky as well. Maybe I'll just go over them. So this disease involves a gene mutation and does not follow an autosomal recessive inheritance pattern. This one is Huntington's disease, and the next one is this disease follows a gene mutation and it does not follow an autosomal dominance inheritance pattern. And this one was Tay-Sachs disease. Okay, and I'll just go on the next screen and just briefly, since we've talked about sickle cell anemia, we've talked about hemophilia, I'll just briefly go over what Huntington's disorder and Tay-Sachs disease are about again. So Huntington's disorder is a degenerative disease of the nervous system that affects muscle coordination and some cognitive functions. Tay-Sachs is a disease that affects young children, results in both mental and physical deterioration, and most children unfortunately die by 5 years of age. Okay, so if you can just remember those definitions for those diseases and also having to do with the dominance and recessive patterns we talked about, then that'll be good. Okay, so we covered quite a bit there, and so I hope your test goes really well. I can't remember, were you going to do your test tomorrow? I can't just remember what day you were going to do it. Okay. If you feel like Wednesday would be better, we can do that too. What do you think? Okay. Why don't you do it Wednesday then? And I will send you out the link for this. It usually takes about half an hour or 40 minutes or whatever to make it up. Actually, maybe I'll just stop it now.